All right. Next macromolecule group that we are going to talk about is the nucleic acids. Now, these nucleic acids you have probably heard about before. You've heard their names before. They are the primary building blocks of DNA and RNA. So it's kind of interesting. You're going to have the monomers, which in this case, the monomers are the nitrogenous base, the sugar, and the phosphate, right? So these monomers are going to make up the larger structure of the nucleic acid. And then the nucleic acid is going to make up the larger DNA and RNA structure, right? So you've got the monomers, which make up the nucleic acid. And then the nucleic acids are going to create the DNA and RNA structure that we know and love so well from our biology classes, right? So let's go ahead and dive in. This is the structure that we're looking at. When we're talking about a nucleic acid, a nucleotide is just a single nucleic acid, right? So a nucleic acid is a single nucleotide base or a nitrogenous base. And these nucleotides are going to be formed of 3 parts, which we're going to dive into in just a second. Quick reminder, I want to remind you what the role of nucleic acids is in your cells. This is going to be something we go into in the next unit in a lot of detail, but nucleic acids are primarily going to be found in the DNA and RNA of our cells. DNA is going to be converted into RNA. RNA is then going to be read by a ribosome, and the ribosome is going to make amino acids. So these are the last 2 groups of macromolecules that we're going to look at. The third group of macromolecules is nucleic acids, and then the fourth group of macromolecules is amino acids. So really interestingly, these last 2 groups of macromolecules that we are looking at are all designed around this idea of making proteins, which are these polypeptides down here. Polypeptides are going to be big old long chains of amino acids which form larger protein structures. So just a quick reminder of that from your general biology courses. Now, the primary structure that we're going to have for our nucleic acids is going to be made of CHNO and a little bit of phosphorus, right? Little bit of phosphorus, maybe usually 1 phosphorus. So this is going to be similar, in fact, to amino acids, but the difference is going to be that phosphorus and the amount of phosphorus, right? So there is going to be a little bit of phosphorus, just that 1 phosphorus found in the phosphate group. Now, nucleic acids are made of 3 structures. The nitrogenous base, this is the first structure. The deoxyribose or ribose sugar, that's the second structure. And then the phosphate group, which is the third sugar, or not the third sugar, the third structure. Now you might be wondering at this point, okay, why is a nucleic acid considered acidic? Because we have a bunch of nitrogen over here, which we know acts as a base when it's in the form of a functional amide group. So what about a nucleic acid or a nucleotide is considered basic? Well, if you notice the phosphate group over here, we didn't really talk about this characteristic of phosphate groups in class, but this phosphate group has negative charges associated with it. This is really important because if you remember when we were looking at carboxylic acid is going to look something like this. Carboxylic acid, after it donates its proton, remember it's going to donate this proton to solution, and it's going to look like this. It's going to have double bond O, C, O minus. This minus indicates that a proton has been donated to the solution. Proton has been donated to the solution. The same thing is happening with the phosphates in this molecule. What we are seeing is, like I said, it's after the Well, I hope you guys can see that. Sorry, my stupid thing was in the way. But a phosphate group, what we're seeing in this picture is sort of after the explosion has occurred. We're seeing a picture after the chemical reaction has occurred. So what this indicates to us is a hydrogen atom was donated to the solution from this oxygen, and another hydrogen atom, or a proton, not necessarily a hydrogen atom, but a proton, was donated from this oxygen. That's how nucleotides are considered bases, is because this phosphate group actually is considered basic. It has the potential to donate protons to the solution. There are 3 different types of monomers that we're going to want to pay attention to. We got the nitrogenous bases, as I just mentioned, the deoxyribose sugars, and then the phosphate groups. Starting with the nitrogenous bases, these are going to be ring structures primarily containing nitrogen, carbon, and sometimes some oxygen, right? Ring structures containing nitrogen as well as some carbon, hydrogen, and oxygen. There are 2 different structures, 2 different larger structures that these nitrogenous bases are going to be a part of. They're either going to be a part of DNA or they're going to be a part of RNA. Again, hopefully you guys remember this from your general biology classes. DNA is going to contain cytosine, guanine, adenine, thymine. RNA is going to contain cytosine, guanine, adenine, and uracil. Uracil and thymine are the characteristics defining DNA and RNA. If you look at a structure and it contains uracil, it's going to be RNA. If you look at a structure and it contains thymine, it's going to be DNA. So we're going to represent nitrogenous bases with just a little b and a circle right now, right? And we're going to draw what DNA looks like. The overall structure, the larger macromolecule we're looking at here is this DNA and RNA structure. So we're going to use a little b to represent our nitrogenous bases. The next thing we need to be concerned with is the backbone of the nucleotide chain. So when you look at DNA, DNA is a bunch of bases, DNA and RNA is a bunch of bases attached to a long chain. This long backbone is going to be made of deoxyribose sugar. If you're looking at DNA, so this is deoxyribose sugar backbone, or ribose sugar, depending on if you are RNA. Now, the primary difference, and in fact, the only difference between these 2 sugar structures is the fact that deoxyribose is lacking an oxygen, that ribose actually has. So if you guys look at this structure, ribose contains the oxygen. Deoxyribose does not contain the oxygen. So that's going to be a defining characteristic of DNA versus RNA as well, is that deoxyribose, the sugar backbone, is lacking oxygen. Now, this is really important. If you remember DNA versus RNA, DNA is going to be found inside the nucleus. It never leaves the nucleus. It's the recipe for everything about a living organism. Ribose is a copy of DNA. So ribose is going to be used once and then thrown away. What do we know about hydroxide groups? We know that hydroxide groups are very reactive. So this gives us that clue as to why ribose sugars, why RNA is impermanent, why it is easy. We can just throw this away. We just keep making it and throwing it away after we use it because it's got this reactive oxygen group. Deoxyribose doesn't have that oxygen, that hydroxide group or hydroxyl group. So the deoxyribose will in fact maintain its stability in the face of potential chemical reactions. So we're going to go ahead and draw this over here. We've got our nucleotide base attached to a pentose sugar. Pentose just means it's a pentagon. It's got 5 sugars. Now, the last piece that we want to worry about is the connector piece between nucleotides. We mentioned phosphate groups in the context of ATP, which is that energy molecule. But adenosine, even though adenosine is not the same thing as adenine, they are not the same structure, it's going to be a similar molecule. It's going to have similar characteristics. You've got the nitrogenous base at the top. You've got a pentose sugar down here at the bottom, and then phosphate groups that get attached on the sides. These phosphate groups, when you add one phosphate group, it's considered adenosine monophosphate. When you add 2 phosphate groups, it's considered adenosine triphosphate. And then when you add, adenosine diphosphate, then when you add 3 phosphate groups, it's going to be considered adenosine triphosphate. Now, this phosphate group is going to be the connector piece between nucleotides. So nucleotides are this structure. They are the sugar plus the base. So if we look at our sugar molecule over here on the left-hand side, we're going to have a phosphorus or a phosphate group stuck to the sugar. That phosphate is going to be stuck to another sugar, which also has a base attached to it. That sugar is also going to be attached to a phosphate group, and that phosphate group is going to have another sugar attached to it, and that sugar is also going to have a base attached to it. This is our basic nucleic acid structure. Nucleic acid structure. You have the 3 components repeating themselves, potentially in hundreds, if not thousands of repetitions. You're going to get the overall, remember the nucleotide is that whole structure, the phosphate plus the sugar plus the base. And then you've got the base, which is sort of that defining feature, adenine, guanine, cytosine, and uracil, right? So these bases over here are going to be adenine, cytosine, thymine, guanine, and uracil. And that's the overall basic amino acid structure. Now, there are 2 different, this is another way to represent this structure, by the way. This is just a picture. This is going to be our base. We've got our base attached to our sugar. Our sugar is attached to a phosphate. The phosphate is attached to another sugar. The sugar is then attached to a base, and so on. This is just a more accurate representation of what I was just describing. Now, nitrogenous bases fall into 2 categories. This is just the base portion of a nucleotide, right? Because if we remember a nucleotide, the nucleotide is the building block of nucleic acids, and it's going to be made of the base, the phosphate, and the sugar. The bases come in 2 different categories. They are going to be either purines or pyrimidines. Pyrimidines have 1 ring, purines have 2 rings. That's the biggest distinction. Pyrimidines have 1 ring, purines have 2 rings. When we're looking at G, A, T, C, and U, these are our nucleotide bases that make up DNA and RNA. Guanine and adenine are going to be purines. Cytosine, thymine, and uracil are going to be pyrimidines. Purines bond with pyrimidines. Purines bond with pyrimidines. So that means Guanine is going to bond to cytosine. So guanine is going to bond to cytosine. Adenine is going to bond to thymine. And adenine is also going to bond to uracil. Okay. So that, I believe that's our last slide. The one thing that I'm going to leave you with is this idea of GCAP puppy. This is just a stupid mnemonic device to remember the difference, or to remember which ones are purines and which ones are pyrimidines. So guanine is going to be a purine, cytosine is going to be a pyrimidine, adenine is going to be a purine, and thymine is going to be a pyrimidine. There's a bunch of these different mnemonic devices on the internet. I highly recommend that you make your own mnemonic device, because obviously we're missing uracil, which is a pyrimidine. So I highly recommend you coming up with your own mnemonic device to remember which ones are purines and which ones are pyrimidines. And with that, we are done with nucleic acids. See you in a bit for amino acids.